So now we can look into the last part, that is the heterocyclic compounds. Let me erase this once again. Yeah, we are having is the heterocyclic compounds. Now heterocyclic compounds are mainly we call them as the nitrogenous bases. Okay, so if you remember the nitrogenous bases, we are having the purines and the pyrimidines, right? We have studied about the purines and pyrimidines in our molecular basis of inheritance, that is the DNA, about the structure of DNA, right? So in the case of purines, we are having adenine and guanine as our examples. And one more we are having is the pyrimidines. We are having the uracil, thymine, and also cytosine, right? Yes. So all of these three belong to the pyrimidines, and only the guanine and adenine belong to the purines, right? So now we are having nucleosides and also the nucleotide. Okay, the ultimate difference between the nucleoside and nucleotide is if the in the case of nucleoside, only a nitrogenous base and a sugar makes the nucleoside. But if you add phosphate molecule, it will result in the nucleotides, right? So generally our DNA molecules and one more we are having is the RNA molecules are made up of the nucleotides only. Okay, so we are having some examples, that is the adenosine, guanosine. Okay, these are all examples for the nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, and also the uridylic acid, etc. are example for the nucleotides. Okay, I have not mentioned it in the presentation, but you can make it out what I'm trying to tell over here, right? So we have discussed the first part of the chapter, and what we can now do is we can look into some of the important questions for the this chapter. The first question what we are having is which of the following is not true? Trichloroacetic acid is used as an agent to grind the tissues. The earth crust and the tissues have same constituents. The tissue contents are only acid soluble. The acid soluble pool has organic compounds. Okay, so you can make it out over here that the trichloroacetic acid is used as an agent. Okay, we can say a buffer or an organic solvent, we can call it in a more appropriate way, for the grinding of tissues. Exactly, this statement is actually true. We use trichloroacetic acid, or in some cases you also use any other complex organic compound for grinding the tissues. Okay, and earth crust and the tissues have made up of same constituents. Exactly, right? Carbon, oxygen, hydrogen, nitrogen. You do find these constituents in both the living cells and also in the case of earth crust. And finally we are having the third statement, that is the tissue contents are only acid soluble pool. No, that is not the case, right? So hence C is our right answer, because you find two fractions, that is the acid soluble pool and also acid insoluble pool. The acid soluble pool is mainly, it's present in the filtrate, and the remaining solid precipitate is present in the muslin cloth, is actually acid insoluble pool, right? You don't find only acid soluble pool, you find two pools, that is the acid soluble and also acid insoluble. The last statement, obviously it needs to be true, that is acid soluble pool has organic compounds. Exactly, it does have the organic compounds. So the right answer, that is the not true statement, is the C option over here. Now the next question, which of the following is least in the earth's crust, right? So now you people have to go to the table and try to refer which of the following is very least in the earth crust constituent, right? Nitrogen, carbon, silicon, and hydrogen, right? The very little constituent what they have mentioned in your textbook is the nitrogen. So hence the right answer is going to be what? Nitrogen, right? Yes, silicon is the second highest constituent, we all know that. So hence the right answer for this question is A, nitrogen. Yeah, so let me remove this. Yeah, but let me mark the answers, C, and here we are having A. Exactly. Now let us look into the next question, that is the third one. Which of the following technique is used for extraction and purification of compounds? Electrophoresis, chromatography, page, that is a polyacrylamide gel electrophoresis. Let me write this: polyacrylamide gel electrophoresis. electrophoresis, right? Now we use this technique mainly for separation of proteins, remember, okay? We mainly use this for separation of proteins. And the last one we are having is the centrifugation, right? So we have mainly mentioned the term purification, though all of the techniques, they are involved in separation and extraction, but the purification is mainly done by the chromatographic techniques only, right? Hence the right answer we are having is B, that is chromatography. Now, the next question. The probable structure and molecular formula can be determined by the physical techniques, chemical techniques, biological techniques, analytical techniques. Okay, so if you look into the previous question, we have mentioned about the chromatography is mainly used for extraction and purification, right? So now chromatography is an example for analytical techniques. Okay, analytical, the name itself indicates for analysis. Okay, and the analysis is mainly used for determining a empirical structure and empirical formula, molecular formula of that particular substance that we are interested in, right? Hence the right answer over here is the D, that is analytical techniques. Probable, what do you mean by probable? Probability, okay? Maybe or may not be, okay? That appears to be true initially, or it may be even false later on, right? Yes. So hence the D is our right answer. The next question, drying and destructive methods such as incineration, that is burning, is used to determine the dash of tissue, that is organic constituents, inorganic constituents, biochemical constituents, all of the above. It is clearly indicative that inorganic constituents, that is sodium, calcium, magnesium, and so on, they are mainly determined by drying the tissue and by burning the tissue. And finally, the ash that remains is mainly used for what we can say, the determining the inorganic constituents. So now let's move on to the next question. The element that is highest constituent in the human tissues and earth crust is silicon, nitrogen, oxygen, carbon. So we have to again look into the chart, and as I had emphasized quite importantly, that oxygen is the highest of all, right? So hence C, oxygen, is going to be our answer for this. That is the highest constituent in both the human tissues and also the earth crust is the oxygen. Now, let's look on to the next question. Which of the following statement is true? Silicon is the least of all constituents in the human tissues, and carbon is the highest constituent in the both earth's crust and also the human tissue. And grinding of tissue with trichloroacetic acid is used to determine the inorganic constituents of the tissue, and inorganic elements belong to acid insoluble. So now let us try to recall all the points that we have made till now. So it's clearly indicative that inorganic elements belong to acid soluble, not for acid insoluble, right? So hence we can say that the D statement is actually false. The next one we are having is grinding of tissue with trichloroacetic acid is used to determine the inorganic constituents. No, right? We mainly use this method for determining the organic constituents. Hence, even the third statement, that is C statement, is also false. The second statement, you can see that the carbon is the highest constituent in both the Earth's crust and human tissues, right? So you can see that it is actually the oxygen that is the highest constituent. Carbon is the second highest constituent only in the case of human tissues, but not in the case of Earth's crust. So this statement is again false. The first option we are having is silicon is the least of all constituents in the human tissue. Exactly, it is the very negligible that they have mentioned in your textbook. So hence A is going to be our right answer for this question. It is the truth statement. The next question, which of the following statement is not correct? The elemental analysis is used to determine the inorganic constituent, and the ash contains the inorganic elements. The functional group such as ketones, aldehydes, etc. are the biological point of view, and carbon derived compounds are called as biomolecules, right? So now again they're asking this time about the false statement or incorrect statement. The carbon derived compounds are called as the biomolecules. Exactly, it is actually true. And the functional groups such as the ketones, aldehydes, etc. are the or in biological point of view? No. The classification based on the functional groups is actually the what we can say biological point of view, but determining the functional groups is actually the chemical or chemistry point of view. So hence we can say the C option is not true. We can directly confirm that C is our answer, right? So hence C is our answer. And the B statement, that is ash, mainly contains the inorganic elements. Exactly, it is true statement. And elemental analysis is mainly used to determine the inorganic constituents. Exactly, even that is also a true statement, right? So hence the right answer for this and the not correct statement we are having is C, or the third statement. The next question we are having, the net charge of this Zwitter ion is 1230. It is simple. We have mentioned in the earlier class that the charge is going to be 0. The net charge is going to be 0. It carries no net charge, right? So hence D, that is 0, is our answer. The next question, the acidic group in the amino acid is the nitro group, carboxyl group, amino group, hydroxyl group. We all know the carboxyl group, that is a COOH. Okay, the COOH, that is the carboxyl group, is mainly giving the acidic group, and the amino group is mainly giving the that is NH3 is the amino group, or NH2 group. It mainly gives the basic property to the amino acids. So hence B, that is carboxyl group, is our right answer. Now moving on, which of the following is an aromatic amino acid? Right? Which of the following is an aromatic amino acid, right? Tyrosine, histidine, arginine, lysine, right? So now we know there are 3 examples that have been mentioned in a textbook: tyrosine, tryptophan, and phenylalanine, right? Only these 3 belong to the aromatic amino acids, right? And hence A is our right answer. Histidine is actually positively charged, okay, and arginine is also positively charged, lysine is also positively charged, right? So all these are actually positively charged amino acid. Now, moving on, match the following. We are having 2 columns, that is column 1 and column 2. The first column we are having is the first one, methyl group, hydroxymethyl group, acidic amino acid, and a neutral amino acid. The column 2 we are having is the glutamic acid. The next one we are having is the alanine, and the valine is the third option. Last we are having is the serine, right? So we all know the methyl group is mainly present in the case of the alanine, so we can write 1 number over here. Hydroxymethyl is mainly present in the case of serine. The fourth option will be having the second one. And the acidic amino acid we are having is the glutamic acid is an example for acidic amino acid. The neutral, the last remaining we are having is the valine, hence the fourth will go over here. So if you look into the options that has been mentioned over here, the first one matches with the second option exactly. And yeah, so we can see that the second option is similar to what we have mentioned over here. B is going to be the right answer. 1 with 2, 2 with 4, 3 with 1, and 4 with 3. Right? Yes. So this is going to be our right answer. Moving on, the next question. Which of the following statement about the amino acid is true? The alpha carbon has 3 different group attached to it. And if the R group is hydrogen, then the amino acid is alanine. Okay, so I think we have missed a word over here, that is amino acid. Okay, the amino acid doesn't change, the charge of the amino acid doesn't change irrespective of the pH in which it is present. I think this is what the option should be. And there are 20 amino acids occurring, right? So they say that there are 3 different groups are attached to it. So we can say that it is actually 4 different groups are attached to the alpha carbon. And if R group is actually hydrogen, the amino acid is called as alanine. No, it is called as glycine, right? So hence a B statement is also not true. And the third statement, the charge of the amino acid doesn't change irrespective of the pH in which it is present. No, it actually changes according to in which pH condition it is present. Even C option is also false. And in the D option we are having is there are 20 amino acids occurring. Exactly, we are having 20 amino acids. So if you refer any higher biology textbooks such as mainly biochemistry and all, they even mention that there are 21 amino acids. Okay, and the 21st amino acid is called as this selenocysteine. which is a modification or derivative of serine amino acid, if I remember properly, right? So I think the right answer over here is the D option, that is there are 20 different amino acids occurring. That is going to be our true statement and the right answer for this question. Now moving on, which of the following is a lipid? Fatty acid, diglycerides, phospholipids, all of the above. Yes, exactly. All of the three mentions are actually the lipids. Diglycerides are also derivative of a lipid. Fatty acid is a simple lipid. Phospholipids are also derivative of lipids, glycerols, etc. Right? So we can say all of the above are the lipids. Now moving on, the total number of carbons in the arachidonic acid is 18, 20, 25, 15. We know in the case of palmitic acid it is 16, and in the case of arachidonic acid it is 20. Hence B is going to be our right answer. The next question, a nucleoside is sugar plus phosphate, phosphate plus nitrogenous base, nitrogenous base plus sugar, and nitrogenous base only. So a nucleoside is mainly made up of a sugar and a nitrogenous base, right? If you attach a phosphate molecule, it becomes a nucleotide, right? So hence the C is, that is a nitrogenous base, and sugar plus a sugar will make the nucleoside. So C is going to be our right answer. The next question, which of the following is not true about lipids? Palmitic acid has 20 carbons. Exactly, it is not having 20 carbons, it's actually having 16 carbons, right? So you can see that A, the first option itself, is not true. So hence A is going to be our right answer. But let us check on to the other three options also. The arachidonic acid is a fatty acid, and oils have a lower melting point, and fatty acids are esterified with glycerol to form monoglycerides. Exactly, D statement is also true, C is also true, and arachidonic acid is a what we can say fatty acid. It is a polyunsaturated fatty acids. So the B, C, D are actually true statements, but the A statement, that is palmitic acid has 20 carbons, it should be having 16 carbons, right? So hence A is our not true statement and the right answer for this question. Now moving on, an example for phospholipid is the fatty acid, gingerly oils. Next one we are having is the palmitic acid. The last one we are having is the lecithin, right? So lecithin is an example for the phospholipids, and lecithin is actually found in the membranes, cell membranes. Remember this point. So D is our right answer. The next question, the lipids with complex structures are found in gastrointestinal tract, neural tissues, sensory system, all of the above. Okay, lipids with complex structures. Example we are having is the ceramide or sphingolipids, etc. Right? So all of them are actually complex lipids, and you have mentioned, like you have read it, that they're mainly found in the neural tissues. Hence B is going to be our right answer. Next question we are having is the DNA and RNA consists only of the nucleotides, nitrogenous bases, the sugars and phosphates, and the nucleotides, right? The first option is nucleosides. Sorry, I mispronounced it. So we all know the DNA and RNA are ultimately made up of the nucleotides only. Hence D is our right answer. Nucleotides is going to be the right answer, right? So let's meet in another video with the next part of biomolecules. Thank you.